Good afternoon. This is Dennis Hooper from Realtime Lab. We are going to have our webinar this afternoon on the comparison of enzyme-linked immunosorbent assays, or ELISAs, with liquid chromatography mass spec in mycotoxin studies. This is who your speaker is today, Dennis Hooper. I'm one of the Realtime Lab principals. I'm also the former medical lab director, but I've now moved into clinical consultant, director of new product development. I've been trained in the U.S. Navy as a pathologist. I retired from the U.S. Navy as a Navy captain, and I have a Ph.D. from the University of California, Davis in microbiology. On the right is my good buddy, Mold Critter, and he is our mascot for Realtime Lab, and he will follow us through this presentation. Why was I interested, and why am I still interested in fungi and their metabolites? I was most interested in looking at the molds or in the fungi when I was in the Navy doing autopsies, and a lot of the transplants or the cancer patients who developed what the internist told us as fungi infections, when we did the autopsy, we found very few organisms. It always intrigued me that how could they die so abruptly and so much in pain when there was, even though they had cancer, but the docs thought that they were dying from the fungi. Well, I started studying mycotoxins at that time and found that they were low molecular weight small molecules. They're usually produced as secondary metabolites by the filamentous fungi. There's 300 to 400 of these nasty little compounds for most part, and they are recognized as mycotoxins, but only 10 to 12 receive attention as human threats. That doesn't mean there's more that shouldn't be looked at. Most of the literature available in peer-reviewed literature attributes mycotoxicosis to ingestion of contaminated foods, but the airborne contamination is a problem as well. We at Realtime Lab look at every patient as a mycotoxin patient if they are positive for those or present with mycotoxins. We don't say where they come from. And you can see the complicated structures of these mycotoxins, and they are associated with human disease, and they cause acute and chronic effects. This is a picture of aflatoxin. There's 4 major aflatoxins. However, there's others that the literature cites, and the ones I've listed here are B1, B2, G1, and G2. These are separate mycotoxins, 4 mycotoxins, because they have different structures than each of the others. B1 is the most potent natural carcinogen listed, and it's produced by Aspergillus flavus, Parasiticus, and others. We all learned that the liver is the primary target organ, but we at Realtime have also found these toxins in lung, brain, and other tissues. Realtime Lab is located in Carrollton, Texas, which is a suburb of Dallas. It's dedicated to the study of fungi and their metabolites, which are called mycotoxins. We began in 2005 as a mycotoxin lab looking for mold toxins. We submitted patents at that time, and we became CLIA certified in 2005, and we became CAP, or College of American Pathology, and CLIA certified and accredited in 2011. At this time, Realtime Lab is the only laboratory in the United States that conducts mycotoxin testing that is CLIA and CAP accredited. We're going to talk about the comparisons of enzyme-linked immunoabsorbent assays along with the mass spectroscopy And on completion of this webinar, you're going to be a participant and you will improve your competency and performance by being able to understand the core concepts of these tests, ELISA and LC-MS. You are going to be able to define the types of samples and materials used best in these different assays. You're going to be able to interpret the results obtained in the two types of assays, and you're going to learn if and how these two assays can be compared, and lastly, to understand the importance of two tests and their relationships to the fungal toxins. At any time during the webinar, you can type your questions up in the area, and we will discuss these at the end. I will be taking all questions and answering them to the best of my ability. I've been told that you should click on the question mark to ask those questions. So we want to know how we're going to compare such different high complexity tests such as the LC-MS and the ELISA. Well, the first thing we got to know is the concepts of how each test works and how they set them up to work. What are the controls and the standards and how are they validated? And what are the regulatory issues for each of these tests? So the first thing we're going to talk about is ELISA. On the left-hand side of this slide, you'll see that the mycotoxin ELISA test tests for, from Realtime Lab, 15 different mycotoxins, not 7, not 4, but 15. Each of these toxins that we test for are different from each other. It's like a family. We have the aflatoxin group or family, and in each one of those there's a B1, B2, G1, G2. There's others, M and P, but these are the major ones that we're going to talk about today. The macrocyclic trichothecene group is listed below, and then there's the gliotoxin derivative that we test for coming from Aspergillus. So to emphasize again, Realtime Lab tests for 15 at the price they charge. They do not test for 7 as previously others have said, and we do not test only for 4. There's a chance that maybe only one of these toxins may be present, and that would make the whole family positive. So we need to be aware of that as we travel through this webinar. On the right-hand side is the concept of how a competitive inhibition mycotoxin ELISA tests work. We first of all make antibodies in an animal that are called polyclonal antibodies. They will recognize specific portions of an antigen. When we make that antibody and we purified it, we then coat it to microwells in an ELISA plate. And these are specific to a specific target. For example, the macrocyclic trichothecenes. The second thing we do is we add sample, which is in this case urine, and we add a conjugate. The conjugate competes with the target substance, i.e. the mycotoxin, which are also in our controls for antibody binding sites. Once that happens, the conjugate and the target substance or the control remains bound to the wells. Then we add a color substrate, and that produces a color change. The results are read visually or in a reader. Now, Realtime Lab only uses a spectrophotometer. We don't look at anything visually, as some people would like to say we do. The less blue color, the more positive it is. And the darker it is, the more negative it is. We use a spectrophotometer that you will see as we go through this that is plotted. The results are plotted on semi-log paper, and we create a graph to read these results. So what are the clinical and other applications of ELISA? Well, you all being caregivers or being interested in medicine have known this test for years. It can detect hormones, serum proteins, infectious agents such as viral, bacterial, and fungal. There are drug markers, tumor markers, immunoassays, and newborn screening. Some of the ones you may be most familiar with are the detection of rotavirus in feces, the detection of hepatitis B, A, and C markers in serum, and detection of HIV and Lyme disease antibodies in blood. However, Lyme disease has moved into a different arena, but HIV, the screening is still an ELISA test. There is also applications in the food industry for allergies, such as milk, peanuts, egg, etc., that people still use ELISA tests for. So what are the standards we use in ELISA? The standards must be purchased as certified reference material, CRMs, when these are available. If they're not available, the standards are screened in a lot-to-lot analysis prior to being put into use. The standards are made to reflect the concentrations of the analyte, which is the mycotoxin, and the level of detection is noted, and a standard curve is created every time Real-Time Lab does an ELISA test. A semi-log curve fit for the standard curve is then used to plot all the standard reading points. And we will not accept a correlation coefficient of less than 95% for any test that's being reported out. The controls are essential. Most ELISA tests use at least two. We use three. We use a high, a low, and a negative. All controls are made in synthetic urine to prevent any misrepresentation on the standard curve from any possible contaminants. So the matrix is not an issue in synthetic urine. All new control lots are compared to old control lots in a lot-to-lot comparison, not only the controls, but the standards and the antibody plates that we are running the test on. So the advantages of ELISA are that the reagents and equipment are relatively less expensive and a longer shelf life than most other tests. ELISA is highly specific and sensitive, especially if you have the specific antibodies that you're looking at. It's rapid and reproducible. There's no radiation hazards which occur during labeling or disposal of waste. ELISA can be used to a variety of infections, which I've already listed. And Real-Time Lab has validated these in various sample types: urine, tissue, nasal secretions, bronchial alveolar lavages, or BALs, and sputums. Of most importance is the tissue, because a lot of clinicians and patients want to go back to a tissue biopsy that they may have had 3 months ago or last week or whenever, or 2 years ago, and we could pull the block from the pathology department and cut it and then extract that tissue with our procedures that we have patented and extract it, and then we do the mycotoxin test. ELISA is rarely false positive because of the interference substance analytical studies we have done in validation. So we're going to come back to more false positive and false negative issues in a moment. The disadvantages of ELISA are few, but there are some. The antibodies need to be polyclonal and recognize the major antigenic component of the mycotoxins, because there's different R groups in each of these structures. The other disadvantage is that there's some people that make monoclonal antibodies. So say we make an antibody to just aflatoxin B1, that's all we have. We can't check to see if there's a G1, G2, B2, etc. So we need to have a polyclonal antibody to conduct a good ELISA screening test. In LC-MS, we're going to talk about how it's done first. Mass spectroscopy was the first issue that was identified, and I've tried to explain it here in that cations and anions are produced. The cations and anions are accelerated in an instrument. The magnetic field is introduced. The amount of bend is measured. The heavier an ion is, the less it will bend. And then the mass charge ratio is then calculated. So on the right-hand side, I have a picture of mass spec. There's vaporization of the sample. Then there's ionization by an electron gun. There's acceleration of focus by electric field. Then there's deflection by a magnetic field. And there's detection by ion counter. All of this is done in a vacuum. Now that's mass spectroscopy as best defined by me and by others. Now we are talking about liquid chromatography mass spectroscopy. If you look at the bottom picture, the liquid chromatography mobile phase is introduced into methanol, acetonitrile, a lot of different substances can be used, or substrates. And then it is pumped through the liquid chromatograph through a column. The columns must have a specific carrier in that, and those columns, depending on what mycotoxin you're looking for, the columns may be different sizes and be different makeup. They go through an ion source, they hit the quadrupoles. Certain ions will stick to the quadrupoles, others will collide and bounce off, and we'll go to a photomultiplier and we'll see those on the graph or on the printout. The standards that are used for LC-MS are internal standards, and this is taken from College American Pathology guidelines. This is what the individuals who will inspect LC-MS will look for. The internal standards are used for recovery variance and matrix effects. So we have a signal that the analyte is putting out, and we have a signal that the internal standard is putting out. We divide those up, and that comes up to the analyte concentration. This should be unique to the test population. Now that test population can be a number of things. It can be the individuals you're testing or the type of area that you're testing in. So that needs to be well defined. You have to have similar retention time as the analyte. That's going through the whole machine. How long is it retained before it is actually read out? Then it has to be added at a concentration that doesn't interfere with the lower limit of measuring interval, which was formerly called the level lower of optical, or of the lower level of quantification. Sorry, I'm trying to think ahead. So the lower level of quantification. The internal standards should not co-elute with interfering substances. They should not share the same mass transitions with others, and they should not contribute more than 20% of the signal for the test analyte at the lower level of measuring interval. The calibrators are important as well for mass spectroscopy. Calibrants should be purchased commercially, and in mycotoxins, they must be certified reference material, a purity of 99.5%. Now, Realtime Lab has tried to purchase certified reference material for years, and we've done a very good job of that. However, at some point, there are some mycotoxins that cannot be purchased as certified reference materials. In other words, the chemical manufacturers run out of the mycotoxins. For those of you who have been with us for a long time, we used to use Roridin A as our macrocyclic trichothazine, and the manufacturers stopped making Roridin A. In fact, they stopped making Roridin E. And I have noticed that Roridin E is present in some laboratories, and they're monitoring for Roridin E. It's interesting. We would like to know where those laboratories are receiving their certified purity materials or certified reference materials on Roridin E. The common problems we get with mass spectroscopy are hydrolysis, oxidation, dehydration, and many others. So they have to be kept very well in a nice dry area. And many calibrators are not available commercially, as I just pointed out in the Roridin E. The limitations of mass spectroscopy when dealing with mycotoxins are many and varied. There's trouble detecting modified metabolites. That means that the human body or the animal body changes these mycotoxins to a different type of metabolite, just a little bit different, that they may add a carbon or a sulfhydryl group or different hydroxyl groups, and it makes them a little bit different. The other problems, it doesn't provide concentrations of the metabolites. In other words, when you are looking at LCMS, we must look at what are the metabolites as well as the parent compound. The LCMS is very sensitive to contaminants, so we must make sure that we don't have alkali metals or basic compounds that are causing interference. There are high chemical backgrounds that defines the detection limits, and this doesn't differentiate specific to non-specific backgrounds. The analyte must be soluble in a liquid matrix, which makes sense. It's not acceptable for multiple charge compounds to be evaluated, especially if they have more than 2 charges. And there are multiple charges on these mycotoxins. Now, let me tell you, we at Realtime Lab have been very interested in LCMS for a long time. We have looked at this with ELISA and compared it. We worked with the University of California, San Diego with Robert Navio, who does a lot with metabolomics, and with Mass Spec. And we were not pleased with our findings at the present time with Mass Spec because of these problems that you see right here, and others that you'll see. So the pitfalls that we noticed when we were studying this concept is that you must define signal to noise ratios. You must have some kind of compromise between conflicting different chemical properties of the analytes. So you must know extraction and how to extract them, and you have to know how they're ionized. Something that CAP looks at very intensely on LCMS is the matrix effects and how certain ions coming from the matrix may suppress your findings in the mycotoxins. Well, in anything, but we're more concerned with the mycotoxin. So there's a suppression and perhaps even an enhancement of the matrix effect. So you need to be aware that if you're having ion suppression in your test, LCMS may call it negative, where the polyclonal antibody in the ELISA may call it positive, and it isn't incorrect. In some or many cases, cleanup still remains required to reduce the matrix effect and increase sensitivity. And there is use of isotopic labeling internal standards in this test. Of interest, there are something that was documented in 2013, but it was documented years and years ago. It's something called masked mycotoxins. Mycotoxin derivatives are found all over the place, and they are undetectable because their structure has changed in human metabolism. They must have specific calibrators to look for the masked mycotoxins. So if I'm only looking for aflatoxin B1, and perhaps there's other mycotoxins, they've been masked for various reasons. There's many in the literature. We can't find them with mass spec, but we can find them with polyclonal antibodies. So always keep in mind, if I'm getting a negative reading on mass spec, do I really have a negative reading, or is it a masked mycotoxin that we're missing? This was first well documented in 2013 in Food Research. This is a fingerprint of how many of the mycotoxins are printed out on a mass spec and an LCMS document. I've also put on the graph structures of aflatoxin B1, aflatoxin M1, aflatoxin B2. And you'll notice that those are labeled in 5, 6, 7, and 8. All right, if you look down on the chart, and if you look at number 5, for example, aflatoxin M1, it has an R group of a hydroxyl group. But what happens if you only are using the parent compound of M1 or B1, and you inject something and you go through the extraction procedures and you don't find anything? So you're misleading the clinician, or you're being misled as a clinician, that perhaps I haven't looked for every toxin. And believe me, there's a lot of them, and it's exciting to look for, but it's also frustrating because you know how many there are that I told you at the very first of this lecture. The clinical laboratory regulations are many and frustrating. So what laboratories can perform mycotoxin tests on human clinical specimens? Clinical laboratories that hold CLIA certificates for high complexity testing can do these tests. CLIA stands for Clinical Laboratory Improvement Amendments associated with CMS and the Human Health Services. But I must remind you that Realtime is the only CLIA CAP accredited laboratory in the USA for mycotoxin testing, and we're very proud of that, and we've worked very hard to keep that accreditation in place. CAP and CLIA laboratory regulations must be inspected at a minimum of every 2 years. CLIA does it, CAP does it. One of the things CAP does is require that we do a self-inspection every off year of our inspection and report our findings to CAP. We can do FDA, both laboratories can do FDA approved tests, but they can also do non-FDA approved tests called laboratory developed tests, or LDTs, all of which these tests are LDTs. They must perform validations on all tests as defined by CAP and CLIA, and they must have the validations on site at all times for inspections. I am well aware of many laboratories throughout the country that have been closed because they have, quote, lost their validations or misplaced them, and it's not good to do that and tell the regulators that. Realtime Lab has gone through 4 of these inspections by CAP since 2011, and we have excelled in each of those inspections. We have gone through many more CLIA inspections prior to the 2011 timeline. LCMS and ELISA testing regulatory concerns are about the same. We're going to go through those. The most appropriate methodology for LCMS, the labs must optimize and validate for the extraction of mycotoxins in urine, because the mycotoxins have different charges. So we have to know the type of solvent used and the volume used. We must overcome the challenge of obtaining acceptable LODs, or levels of detection, and levels of quantification to achieve the desired sensitivity. So we have to use parent toxins to evaluate these efficiencies. And we have to ensure that we are finding, or at least feel more comfortable that we're finding those masked toxins in every way that we can work on. This was well documented in a paper that was just published in 2015, and I've given you that reference below. Solvents and their effects on mycotoxins are important as well. In studies conducted with acetonitrile, with and without sodium chloride, ethanol, and chloroform, there's different results obtained with different mycotoxins. Another frustrating thing I found with mass spec when we were working and looking at these in UCSD. Enantitin A and zearalenone and aflatoxins have much different findings dependent on the type of solvent used for the extraction. So as the clinician, we need to know what kind of solvent is being used because each of these are different. Acetonitrile is best with zearalenone and enantitins, and aflatoxins have different results in ethanol, acetonitrile, and chloroform than zearalenone and enantitins. The extraction recovery related to polarity index of the solvent is extremely important and is some question that CAP asks in their evaluation of the validations. There are class-specific methods to improve the sensitivity through the LCMS. The regulatory issues for ELISA, there's many, but the most important two for equipment is the spectrophotometer checklist and the pipettes. You must have good pipetting skills and you must have good pipettes. The validations are the same for any LDT, for ELISA or mass spec. They check for analytic accuracy and precision. They check for analytic sensitivity. They check for interference in the interfering substances. If you're going to do quantification, you have to have reportable ranges and reference intervals. These are very important when you work with the FDA because these are issues that when you're doing quantification studies, you need to have a clinical trial to report these reportable ranges. And there's a method validation approval that has to be done and signed off by the medical director or lab director. So the summary of compliance issues are, as you can see, interesting. Laboratories inspected by either CLIA or CAP are subject to strict compliance and must have validated documents available at any time for review. The test results and reports. Now I'm only going to go over the ELISA report because I'm not familiar enough with mass spec reports that are coming out of various laboratories. Difficult to understand, to say the least. If you can look at the top of our ELISA report, we report out the tests that we conduct, ochratoxin A, which is 1 mycotoxin, and then aflatoxin group, which is 4 mycotoxins. Trichothecene group macrocyclic is 9 mycotoxins, and the gliotoxin derivative is 1 mycotoxin. We have reported, and in our validations, we have a level of detection, and that's what we label as, if we meet that level of detection or over it, we sign them out as present if it's greater than or equal to that. If it's not present, it's less than another number, which is, let's take ochratoxin, 1.8 parts per billion. The equivocal range we have marked in there is 1.8 to 2.0. Each of these are statistically determined by the numbers of tests that Realtime Lab has done over the years. Now, a lot of people ask, well, why do you have an equivocal range? Well, why do we have an equivocal range in HIV? There's certain areas that it's questionable, is this positive or negative? I'm sure if you're taking an HIV test, you would rather have an equivocal than be signed out as positive for sure, and then be found out later you are not. So if we get an equivocal, we then sign them out as equivocal, and in talking with the clinician, we suggest that they retest in another month to 2 months. Down below, you'll see our graphs that we have put on our results, and these reflect a scenario that you'll see at the bottom of the slide as well. We test the ochratoxin. Let's just take ochratoxin again, and this tells the dates of the patient's test and retest. And we highly recommend during treatment that the patient is retested, and we test for a lower price on those whole groups of toxins. So the perfect scenario would be a baseline test, and it can be very small to even negative. But if the symptoms are there, the docs say, well, we need to do a challenge, or we need to do something to see if we have the toxin. Once we find the toxin, they continue to treat, and it forces these mycotoxins from the inside of the cell, or these are intracellular, and they're affecting DNA and RNA and protein synthesis. They're forced out of the cell, and at a certain point, we reach a peak of treatment, and then it progresses downward. And we have seen this in hundreds of patients here, and it goes along with symptoms that the docs report to us. We've done many observations with clinicians, infectious disease people, and family practice docs, as well as integrative medicine. And this peak is very common in all the individuals we talk with. The mycotoxin testing advantages by ELISA. Mycotoxin load is reported in parts per billion of urine, providing greater sensitivity and specificity. A lot of documents and a lot of statements have been made saying, why do they do it in parts per billion? Well, when I was looking at this test in 2004 and 2005, I noted that the USDA and other groups in the federal government were reporting mycotoxins out in parts per billion. So it was best that we correlate that to a known test. I talked with FDA about this, and they suggested signing them out as parts per billion until who knows when. Okay, this is performed as a semi-quantitative test that provides clinicians and physicians with a positive or negative answer. So it's present or not present. And we give a numeric value in a semi-quantitative test. We can do that to track patients' progress. That's the only reason we give that. We don't give it to say, oh, you're in the danger zone or you're in the good zone, because it's our belief that if a patient has symptoms and if a patient has mycotoxins, we want them out of the body. We have many, in fact, over 50% of our population that is sent in is negative for mycotoxins. So there's other things that they're looking for, or that they're sick with. This test is reliable and scalable. It's easily adapted to automation. It's cost effective compared to other technologies. The common technology used in clinical industry, leading to a relatively short training cycle with technologists, involves ELISA. And the various specimen samples that we have, urine, nasal secretions, tissues, bronchial alveolar lavages, or BALs, are validated in Real-Time Lab. So why choose Real-Time Lab? I've gone over these. Accreditation. It's registered and accredited by clinical laboratories under CLIA, Improvement Amendment, and the CAP, College of American Pathology. It follows up on testing programs. It has a successful protocol that doctors use that take time, and often several follow-up tests are needed to track the efficacy of that treatment plan. Now, something that you may see in Laboratories is, well, we are involved with CAP proficiency testing. Well, as a CLIA CAP laboratory, you must be, you're not voluntarily giving or agreeing to take a proficiency test. They tell you if they're available, you have to take them, or you have to create them yourself and do them 2 to 3 times a year. So that's how proficiency tests are done. In CAP, they send out a certificate saying you participate with CAP, and then in small letters it says proficiency testing. So it doesn't mean that they are accredited, they are participating with a CAP proficiency test. We also involve ourselves with direct access testing, so the patient can actually call in or go onto our portals, and there's 26 states and the District of Columbia that now allow the patients to order their own lab tests. Well, a lot of issues are involved here, because why do we want the patients to order their tests? They don't know what to do with them when they get them. So when they get them, they call the laboratory here. Our clinical consultants give them suggestions on what physicians to see who know about mycotoxins, and we usually give 2 or 3 in an area that they're interested in. We're very pleased that we have accepted Medicare, and Medicare has accepted us, and TRICARE patients on various tests on this menu. We can also file the claims electronically on your patient's behalf. We don't take any money from the insurance company. That goes directly as an EOB to the patient. Patient navigation is something that we're very proud of, in that we have qualified RN support. Our client services division has a department that is very dedicated to client services and assisting doctors, office help, and patients with questions regarding their specimens and results and billing. And lastly, they have direct access to a medical team. Our medical team can provide expert witness testimony on numerous occasions, which we have done, and they are available to discuss test interpretation, symptomology, and treatment. In fact, in the fall of this year, Realtime Lab is having its 3rd summit here in Dallas on August 3rd and 5th through 5th on treatment of mycotoxins. And we have a number of prominent speakers who will be talking about their treatment plans. We have selected individuals who have published, who are well respected in the area of their treatment plans, and we're looking forward to that. So you can watch our website for any information on that. So summary points. This is my summary of ELISA and mass spectroscopy. I'm going to go through these one at a time. ELISA, well characterized in peer-reviewed literature. This type of testing is accepted and approved by the FDA. HIV, hepatitis, the galactomannan test, which looks for Aspergillus proteins on the outside of an Aspergillus, just a few to mention. And mass spec technology is not approved by FDA for toxins. The only thing I can find is that it's accepted for vitamin D assay by SCIEX Diagnostics. Now that doesn't mean that mass spectroscopy isn't good. It's just questionable in my mind for mycotoxins. Number 2 under ELISA. Technology is accepted and approved by the USDA for mycotoxin evaluation. Hence, that's where I got the parts per billion. On mass spectroscopy, technology is not approved by the USDA for mycotoxin evaluation. However, when you read the literature, you will see that the technology for foods is looking for mycotoxins in mass spectroscopy. But I'm very concerned that they're missing those masked mycotoxins when you look for that in food. Maybe they have a technique but I have not been able to read the literature and find that. Number 3 under ELISA, uses monoclonal or polyclonal antibodies for detection. These cross-react with many in-kind mycotoxins, i.e. the macrocyclics. I gave you about 9 of those in that family. There's many more. And then most antibodies are proprietary in nature. Ours certainly are. Under mass spec, there's no certified material available for many parent compounds, as I pointed out in Rorden E. Maybe there is. I'd like to find out where it is. Who's hiding it? And any other modified standards, because if they modify it at all, then it's difficult for mass spec to find it, and from what I have seen so far. In that the ring forms may change, the carboxyl groups, sulfhydryl groups may change, and we need to identify all those related mycotoxins, because we're misleading the clinician if we're saying that there is no mycotoxins. In other words, ELISA is calling them falsely positive. The 4th issue is CLIA and CAP compliance issues are met in all chemistry and immunology areas. So when we're inspected in chemistry and immunology for under CLIA and CAP compliance. Under mass spec, compliance issues involve limitations for all parameters, which include ion ratios, retention ratios, you can see the analyte suppression causing problems, and all of these can lead to false negative results. So as long as the laboratory who's doing that can explain that they have worked around all these issues, then they have nothing to be concerned with regulatory. Again, here's my buddy. He's reminding you that if you have questions, you can certainly type them in now, or you could call me, or you could write to me at my email at any time. You can find that on the website, and ask questions, or call us and ask. We really respect the people who are working in this area. We are not trying to say that mycotoxins, there's a issue with mycotoxins that only ELISA can do. But we need to be aware of all the problems that occur in both these testing procedures. So the first question that I have is, shouldn't all calibrators for all assays be CRM purchased commercially, regardless of technology? Aren't all calibrators, regardless of tests to be used, supposed to be stored properly? Okay, so there's 2 questions here. Shouldn't all calibrators be CRM purchased? Yes, but there are issues that they aren't available. So you have to ensure under CLIA and CAP regulations that you have done all the types of assays necessary to prove that you're associating these with the right CRMs. Your second question is, aren't all calibrators, regardless of tests to be used, supposed to be used and stored properly? Of course. I hope I didn't mislead anybody there. Okay, why is it, the next question, why is it not good enough to detect the parent compound? Well, I think it was obvious in my discussion. The parent compound is very good to detect. However, if you have a kid or another product that has a different carboxyl group, he may migrate down the path a little bit, or he may have a different group, a larger compound on that structure, and will mislead you to think that you don't have that aflatoxin. If aflatoxin is, here's the next question. If aflatoxin is detected by a masked metabolite, wait a minute, but a masked metabolite is not detected by LCMS, how does this change the clinical interpretation if qualitatively the patient is still positive for aflatoxin? There is no, you're exactly right. If aflatoxin is detected, if you find a B1 and you report it out, that's fine. But what happens if you don't find anything and you're comparing to ELISA that has all the antigen or the large antigen with all the changes noted, then you're misleading the clinician again. Not intendedly, but you've said there's no aflatoxin if you're only using one parent compound. Next question, how is the requirement for a mycotoxin being soluble in the LC-MS model mobile phase different from it needing to be soluble in ELISA assays? It isn't. It's the same thing. We have the same issue. It has to be soluble in our ELISA assays, or we're testing, our test is for naught. These are very good questions, by the way. How are ELISA polyclonal antibodies more specific than detection by LC-MS? I think I've explained that in that the ELISA polyclonals they recognize the whole family. The LC-MS is looking for the child or the parent, but only one component. Next question. Can you add other foodborne mycotoxins like DON, fumonisin, zearalenone, since those are common contaminants? We can, and we are working on many of these now. I've been told by marketing here that until it's ready for prime time, I can't say. But yes, I agree that we need to look for more. DON is a simple trichothecene, group A, and we could certainly look for that in the ELISA test. And I'm sure that they could look at it through the mass spec. However, DON also has there's a T2 and a HT2, so we have to be careful of that. Is the environmental analysis different from the clinical? This is a little bit more difficult because we get different assays from the environmentalists, and we look at dust, we look at swabs, we look at different components of the environmental areas. We extract it through our extraction procedures, and we analyze it in the same manner as we do the ELISA test for urine. And we use the standards and controls in the same way. So it is, in some ways it's different because we use a different component, but in many ways it's very similar. You mentioned a challenge test. What is utilized for that challenge test? Okay. I wish more people would publish in this area because it's an exciting area to look at. I get many calls saying, how come my patient is negative with your test and with mass spec, and I just know they have something wrong with them? Well, some of the docs that we have had for years have done challenges, much like cortisol challenges that we're familiar with in medicine. And that is that they give them a glutathione challenge, either IV or liposomal glutathione, 2 teaspoons twice a day for 3 to 6 days, and then they retest. Or, interesting, is they put them in saunas. And when they start going through saunas, the cells change and release these mycotoxins. And we find that we get those curves. And then as they progress through a treatment plan, they become more negative. So the challenge test needs to be published, and I've been pushing people to do that. Okay. Where are the certified reference materials for ELISA? You can buy them through Sigma. You can buy them through Enzo. These are chemical companies. Next question. If a lab has chosen a certificate of registration and achieved a certificate of compliance by CLIA How is that lab different from a lab that has chosen an accreditation route through CAP? You might want to ask CAP that, because CAP emphasizes accreditation, and CLIA has an accreditation, which is the highest CLIA certificate that they can give. Certificate of compliance is the second one that CLIA gives. So there's a reason why they do that, and I think, personally, I think it's just because they have more questions and more certification hurdles to go through. I don't know that, and it's a question for CAP. Next question, is it impossible to test other sample types by LC-MS? I believe not, but I don't know for sure. If they do, they need to have it validated. I have done inspections of laboratories that are CAP accredited, and every time, and these are all LDT tests, and when I look at those, they have to have their validations for each sample, just like we have to have. So it's no different. Next question, how are ELISA polyclonal antibodies more, oh, we already had that question. We're going the wrong way. The masked mycotoxin issue seems to be related mostly to metabolites formed by DON in certain animals and then ingested by people. Are there other masked mycotoxins? That's a good question. I don't think enough work has been done on that to tell. But are you going to feel comfortable signing out a case if you have said it's negative and you don't know if there's masked mycotoxins? I would refer you to the papers I suggested and do a clinical search on those papers. And then if you want to, we can talk about this at a later date. I think it's a good area to investigate, but I can't answer that question. That's all the questions that have been submitted, and I appreciate your responses, and please follow us on realtimelab.com, on our Facebook and our Twitter. And I look forward to doing more of these, and if you have any responses, please let me know privately or any way you want, by our email or by my phone call. Thank you again, and good day.